that you sent out an email with the extra credit, but I shot you an email, but I'm not sure if anybody else, I never got the worksheet for the extra credit.
All right, I've got a link for it up in Canvas now. Let me see if I can show you that. And I just got the link last night, so I extended the time by a week that it has to be turned in. Where is that? There it is. Can you guys see my screen?
No.
No.
All right. There we go. Now can you see my screen?
Yes.
All right, so if you click here, extra credit study sheet, this will open up, and then you click here, you'll download a log, which will be a spreadsheet, and let's open it. On the first time, put your schedule, like when you're working, when you're planning to eat, when you're planning to go to bed. And if you want to be somewhat private about this, don't put in direct information like, I don't know, you don't have to put in that you're going to the shopping at Safeway. You just say something like shopping, okay? Anyways, oops, that's the wrong one. Schedule there. So it says on the top how to do it. On this worksheet, import your schedule. Do not put your study times. On log 1, put your study time for week 1 or week 2, because week 1 is almost gone. And all other weeks, it's the same. If you're planning to use this same schedule, the study schedule, for all term long, or all semester long, sorry, then that's all you need. However, in week 2, let's say you're going to go visit, I don't know, a family member out of state, and so that'll impact your study schedule. So here, on log 2, put your study schedule for week 2, and then for week 3, put it on log 3, and for all other weeks, if it's different, you just click this button to add another page. Any question about that? Most of you will probably be doing the same study schedule for all semester long, in which case you'll only need one. But if you need more than one, that's fine. And put on here when you're studying for microbiology. Like if you're taking another class, I need to know when you're studying for microbiology. The other class, you can put when you're studying for that other class. I don't need to know that, but I do need to know for when you're studying for microbiology. And then I'll compare that with your schedule to make sure you can actually do it. And if you're not studying enough, I'll just give you a comment. Somebody turned in a schedule to me, and they had scheduled something like 4 or 5 hours of sleep a night. And I said, is that right? You're only going to get 4 or 5 hours of sleep? So I will take a look at it. Anyways, if you turn it in, and the due date has been moved from this Saturday till next Saturday, because I didn't get the link up until last night, I'll give you 6 points of extra credit. So all it is I want to know when you're planning on studying. And this will be good for you because it'll get you thinking about studying for the semester. Any questions about any of that? All right. Were there any other changes I made? I'm not sure I mentioned it last time, so let me mention it this time. I did changed the lockdown browser, so it should work. I think I did mention that apparently Lockdown Browser is going to be set up for each college, and I didn't know that. I thought it was just a link to download it. And I think I told you that the length of the class is correct on Tuesday and Thursday. We're going to have a very long class on Tuesday and Thursday because we don't have a lab. And I will give you a break in that schedule. And you guys can think about, do you want the break in the middle so you can get something to eat, go running and do something? Or would you just like to leave the class early? Let me know. And we will talk about that next time. All right. I have one other change I want to make in the Canvas website. I'm not even going to tell you about it, but it's near the end of the term. But I think the website is pretty much finalized now. I don't believe I have the lab up past week 4. Let me check that. And that's because I don't want to overload you. But I will put it up when we get closer. But it's up, you just can't see it. And I just don't want to overload you. All of the lecture is up. So week 8 of lecture has the final up there. Any questions about anything? All right. Let's take a look at what we're doing. We're working on Chapter 1 today, and hopefully we'll finish Chapter 1. Later today we will be doing Lab 01, and that will be at 2:30. Any questions about the schedule? If not, let's go on to our lesson. Now you see why I like to put 2 here when we end. All right, I think I talked about this slide. So let's move to the next slide. Fermentation and pasteurization. You don't need to know when, but Pasteur in 1857 showed that microbes are responsible for fermentation. He showed that alcoholic fermentation occurs from the conversion of sugar to alcohol by the microbes, and this is how we make beer and wine. Surprisingly, before that time, people didn't know that. Microbial growth is also responsible for the spoilage of food, and Pasteur actually showed that. And what he was doing was he looked at the beer and wine industry, and he looked at why the beer and the wine sometimes went bad. And he showed that the bacteria that make the alcohol can have another bacteria that comes in it and then can use up the alcohol, producing acetic acid, spoiling the wine by fermenting it further to vinegar, meaning converting the alcohol to acetic acid. And of course, if you drink that, that doesn't taste too well, and they considered it spoiled. Pasteur further went on to show that the spoilage bacteria in the beer could be killed by heat, and he applied a heat that was not hot enough to evaporate most of the alcohol in the beer and the wine. And he applied this for the beer and the wine industry. So pasteurization came about because the beer and the wine industry wanted to stop their product from being spoiled. And then pasteurization became available for a large number of other products, and it's mostly used today with milk. Pasteurization is the application of a high heat for a short period of time. It's used in a wide variety of foods. Did we talk about this last time? All right, I guess not.
I think you touched on fermentation, but I don't remember you in this much detail.
Okay. Thank you. Fermentation, the realization that the activity of microbes could affect an organic matter, like grape juice, converting it to beer or wine, which I guess if it's grape juice, it's converted into wine, and then the microbes converting that wine into vinegar, led to the realization that microbes could have similar relationships with plants and animals. And could microbes cause disease? This concept became known as the germ theory of disease, that microorganisms, or germs, cause disease. Originally, before the germ theory of disease, it was believed that poisonous vapors were responsible for causing infectious diseases. Like the reason why a person got cholera was because they were walking through a swamp. It wasn't because in the swamp there was the microorganism that caused cholera. They thought it was the poisonous vapors. And that was just the medical thinking at the time. With the germ theory of disease, there's a number of people who advanced it. Semmelweis, advocating the washing of hands, and Lister, using a chemical disinfectant, which was carboxylic acid or phenol, to prevent surgical wound infections. Anyways, the idea was that germs cause disease, and then this was finally proved by Robert Koch. This is another name you should know because it's one of the founders of microbiology. And you don't need to know when he did it, but in 1876 he proved that a bacterium, Bacillus anthracis, causes the disease anthrax. So you should know that Koch showed that Bacillus anthracis caused the disease anthrax. Koch then wrote up his experimental steps to prove that a specific microbe causes a specific disease, and these became known as Koch's postulates, the steps you use to show that a given organism causes a given disease. We'll talk about Koch's postulates 3 times during the semester. So this is one of those major topics that we'll hit, well, in 2 other times. We'll go into it in much more detail the next time I talk about it. Anyways, Dr. Koch then proved the germ theory of disease, where a given microbe causes a given disease. And like I said, he formalized his postulates, and they became known as Koch's postulates. Any question about any of that? About this time, we had the first vaccine, and that was developed by Edward Jenner. He made the first vaccine against smallpox by inoculating a person with the cowpox virus. At the time he made this vaccine, nobody understood why it was working. They didn't know much about the immune system, and they did not know how vaccines worked and then provided immunity. But the person was protected against smallpox by the cowpox virus. So this is another virus. This was not understood until Pasteur showed why it was working. And with Pasteur's work on vaccines, it was then possible for other people to make other vaccines. So Edward Jenner, when he made his vaccine, nobody knew how to make a vaccine. Is that clear? And it took almost 100 years before people understood what was going on and how to make vaccines. And it was Pasteur whose work showed it. He was actually working in the chicken industry, trying to prevent a disease caused by a virus in chickens. And he worked on it, and he showed that the reason why the cowpox virus was protecting against smallpox is that the two viruses were similar to each other. And then when the immune system responded against the cowpox virus, it also responded, at least part of it, against the smallpox virus, because the smallpox virus was related to the cowpox virus. Any questions about that? The term vaccination is derived from the word vaca, which stands for cow, after the cowpox virus. Any questions about that? So vaccination comes about because the first vaccine was the cowpox virus. And the protection we get from a vaccine is called immunity. We'll talk a little bit more about vaccines when we're talking about viruses, and I'm talking about COVID-19, but generally we won't be talking about vaccines. Any question about any of this? So with Louis Pasteur's work, we then got the concept of chemotherapy, that it might be possible to treat certain diseases with chemicals. And a chemotherapeutic agent is an agent that's used against an infectious disease. It can be a synthetic drug or an antibiotic. So when we use the term chemotherapeutic agent, in this sense we're meaning an antimicrobial agent. But you should realize that chemotherapeutic agent is any agent that is used against a disease. And generally, when we use the term chemotherapy, we're referring to a treatment against cancer. But the term can be used more broadly to include, like, treatment with antibiotics against a bacterial infection. Any question about any of that? All right. So an antibiotic, by the textbook definition, is a chemical that is produced by a living microbe, either a bacteria or a fungi, that inhibit or kill other microbes. This is the textbook definition. Now you should understand that most people, as well as most clinicians, do not use the textbook definition of an antibiotic, because by the textbook definition of an antibiotic, penicillin G is an antibiotic because it is produced by a living organism. However, ampicillin, which is one of the early members of the penicillin family, is not an antibiotic because it is actually made by a living organism, but then humans chemically modify it. And that would be called a semi-synthetic. You don't need to know that term. And then there's some some chemotherapeutic agents which are totally man-made. I think carboxylin is a totally man-made member of the penicillin family, meaning humans make it in a lab. Now, all of these are members of the penicillin family, and it doesn't make a whole lot of sense calling one member an antibiotic and another member a semi-synthetic, and then the third member a man-made product. What do they call that? A synthetic chemotherapeutic agent. As I stated, the term antibiotics, used in general by the population, as well as most clinicians, is to include any chemotherapeutic agent that can be used against bacteria. And that's the way I will use the term antibiotic. Meaning, when I say carboxylin, I will call it an antibiotic. Any question about that? All right. The first chemotherapeutic agent that was available to treat a disease was quinine from the bark of the cinchona tree. And it was the first drug available to Europeans, and it was used to treat malaria. Anyone want to guess when the first drug was available to treat malaria? We'll say what time frame, like late 1900s, early 1900s, early 1800s, something like that.
Was it World War I? 1930s, maybe? I don't know.
No. Anyone else want to guess?
Wasn't it like in the mid 1600s? Because
Oh, somebody knows, yes. It was around the 1650s. They're not entirely sure exactly what year because back then the records weren't that good. But it was actually Jesuit missionaries in Peru noticed the natives eating the bark of this tree to treat their malaria, and noticed that it was successful. And then they took the bark of the tree back to Europe. And so the bark of the cinchona tree, which had quinine in it, was used to treat malaria way back in the 1600s. And that was the first chemotherapeutic drug that the West had. Now, in China, there was the drug they had also for treating malaria, and I think it was artemisinin. And that happened in a long time ago in China. I'm just going to say it that. What's surprisingly, this drug was effective, but the Chinese forgot about it. Maybe there was a revolution and new leaders came to power, and I don't know if they killed off the medical establishment or what, And so China forgot about this drug that was effective, and they rediscovered it, I think, in the 1960s. Any question about any of that? Anyways, from largely Louis Pasteur's work, but also Dr. Koch's work, it came to the concept that we might be able to find a drug to treat diseases, infectious diseases. And Paul Ehrlich was a proponent of this. He was talking about finding the magic bullet that could be used to treat infectious diseases. And in his lab, he just started applying different compounds to try and treat the disease syphilis. He was able to infect rabbits with syphilis, and then started using different compounds on them to try and treat them. And after he sampled about 500 different compounds, he found a magic bullet, the first drug to treat syphilis in about 1910. It was a synthetic arsenic drug, salvarsan, that was effective at treating syphilis. The trouble is, if you don't know, arsenic is, highly poisonous to humans. And so, although the synthetic arsenic drug did successfully kill syphilis, it was very hard on the patient, and you had to be very careful or else you would kill the patient. Any question about any of that? So in 1910, we got the birth of modern chemotherapy, where we found the first drug that could cure syphilis. And this is when people were actively looking. Obviously, there was a drug available to cure malaria way back in the 1650s. In 1928, Alexander Fleming discovered the first antibiotic. He was a microbiologist and a doctor, and he was streaking out his bacteria, and he was a sloppy microbiologist, and contamination got in the plate. Furthermore, he left the plate on the counter and then went away for the weekend and came back. He left the plate on the counter instead of putting it in the incubator, is what I guess I should say. And at room temperature, both the bacteria and the contamination were able to grow. In the incubator, only the bacteria would have been able to grow because it was a higher temperature. When he looked at his plate, he noticed the bacteria were growing and that he had contamination on the plate. But when he looked at the contamination, he noticed a ring around the contamination where there was no bacteria growing. And he correctly concluded that something was diffusing out of the contamination and preventing the growth of the bacteria. And he called this an antibiotic. It actually was the first penicillin that was discovered, penicillin G. And he called it penicillin because it was made by the Penicillium mold, a fungus. Any question about that? Sometime later in the term, I'll
Where
talk
does
about...
penicillium find?
Can you repeat that? It broke up, so I didn't hear most of that.
I'm just wondering, is that like a mushroom or something?
No, it's a fungus, a mold. It's right there, a fungus. It's a colony of mold got established on his plate because he was a poor microbiologist, and when he streaked out his plate, it got contaminated. You probably left it open to the air too long. All right, later in the term, remind me, and I'll tell you the story of the first 2 uses of penicillin G, in the clinic. I don't have time to talk about it now, but I'll talk about it later because it's an interesting story. Surprisingly, penicillin G was not the first antibiotic to become clinically available. In Alexander's day, way back in the 1920s, he could not purify penicillin. All he could get would be a crude extract of media that he could filter out the mold from it, but you can't inject media into people. It wasn't until much later that the biotechnology advanced, and then they were able to purify penicillin and then use it as a clinical drug. So penicillin was actually the second antibiotic that became available to the clinicians. It was the first one discovered, but the second one that became clinically available. And that's not a major point, I'm just informing you. The first antibiotic, quote unquote, was a sulfa drug, sulfonamide. And this was a man-made drug, so specifically if you're using the textbook definition of an antibiotic, the sulfa drugs are not an antibiotic. They are a synthetic antibacterial agent. Because it was man-made, it could be man-made in a pure form, and it became clinically available right before World War II, about 1935. So the sulfa drug was the first drug, or the first quote-unquote antibiotic that became clinically available. The penicillin was clinically tested and mass-produced in the early 1940s during World War II. Any question about any of that? And I'll give you the story of the first clinical use of penicillin. You might have to remind me, but I'll do it later in the term. When we talk about microbes and they're causing human disease, you should realize that bacteria were once classified as plants, hence the term flora for microbes. And if you hear me ever use the word microflora or flora, I apologize. That was the term that was used for bacteria when I took microbiology. And that was ages ago, right? That term is no longer used because we don't classify bacteria as plants. And we haven't classified bacteria as plants for a long, long time. It was at least the 1950s, if not earlier. We have replaced the term microflora with the term microbiota. And when we use the term normal microbiota, we're referring to the microbes that are normally present in and on animals and plants. And when we're talking about the human normal microbiota, we're talking about the microbes that are in and on a human. Usually when I'm talking about the normal microbiota, I'm talking about the human normal microbiota. But you should realize that all animals and plants have a normal microbiota. And those are the microbes that are living in symbiosis with the host. And there's a whole community of microbes that are living in symbiosis with the host. And they're actually central to the host's welfare. Any questions about any of that? So you need the normal microbiota to stay healthy.
I have a question.
Uh-huh.
What is it called when your normal microbiota becomes, I don't want to say pathogenic, but for example, small intestinal bacterial overgrowth, where they say, something that's normally present in your large intestine maybe moves up into your small intestine and multiplies rapidly, and it shouldn't be there, and it's not normal. What is that called?
That's called an opportunistic infection. We'll talk about that term later. That's where the normal microbiota causes an infection in the patient, usually because it's growing where it shouldn't be growing. But good question. All right, let's talk a little bit more about the normal microbiota. The normal microbiota are found on and in the skin, the oral nasal cavity, the respiratory tract, the digestive tract, the entire gastrointestinal tract, and the urethra. The normal microbiota is not normally found in the internal organs and tissues. So the normal microbiota is not in the blood, an internal organ like your heart, your muscles. But it is on the skin and all of the mucous membranes lining the internal cavities of the body. The upper respiratory tract has the normal microbiota, but the lungs, to my knowledge, usually do not. And if we get something growing in the lungs, it causes pneumonia. The urethra has normal microbiota, and I will say, at least in women, we'll talk about that a little bit later, the human bladder, at least in women, has a normal microbiota. The studies I've read have only looked at women, and so I don't know if the normal microbiota is in the male bladder. The normal microbiota, excuse me, the normal microbiota prevent the growth of pathogens. How do they do that? They compete for resources and for space. For example, if I were to get a pathogen on my skin, the pathogen may not find space to grow. It may not find resources either because the normal microbiota on my skin is already there. And that's one of the reasons why you need usually a large number of pathogens before you'll come down with the disease. Even salmonella, you need something like 500 cells before you'll come down with salmonella. And it's because of the normal microbiota preventing the growth of the pathogen. Some normal microbiota can actually secrete toxins that inhibit the growth of pathogens. And we'll talk a little bit more about that later in the term. For example, some of the bacteria in your mouth prevent toxins that inhibit the growth of pathogens. And the E. coli in your gut can produce a toxin that inhibits the growth of a pathogen, like salmonella. So if the salmonella gets near that E. coli, it's not going to grow. Some of the normal microbiota produce nutrients and growth factors for us. For example, humans get a significant amount of folic acid and vitamin K from our normal microbiota. And then our normal microbiota also help us in digestion. E. coli helps us digest our food. A classic case of the normal microbiota helping digest food happens with the cow. The cow cannot digest grass because grass has cellulase in it. And it's the normal microbiota in the stomach of the cow that digests the cellulase. The cow cannot digest grass. Any question about any of that? All right. The normal microbiota do not normally cause disease. However, they can cause disease in certain circumstances. So normally, the normal microbiota do not cause the disease. But in immunosuppressed patients, the normal microbiota can cause disease by growing in places where it shouldn't. Another example of the normal microbiota causing disease is a yeast infection. We have yeast as part of our normal microbiota. But when we lose bacteria that compete with the yeast, like when you take a dose of antibiotics, it kills off the bacteria, but not the yeast. And then the yeast can bloom, and it might cause a yeast infection. Any question about any of that? All right. An infectious disease is one that results when a pathogen overcomes the host resistance. And this would be by a bacteria that we call a pathogenic microbe. What? Sorry, I don't know why that ran. Did you guys hear that? As well as see it? All right. An infectious disease is caused by a pathogenic microbe. An emerging infectious disease, or EID, is a new or changing disease that are increasing or have the potential to increase in incidence. what is a new and emerging infectious disease that we're facing right now? Come on.
COVID-19?
Yes, COVID-19 is a new and emerging infectious disease. Hopefully, it's going to be decreasing in the future, but in some places in the world, it is increasing.
I have a question about that though. Hasn't COVID-19 been around for a while though? Or
I'll
that
talk about that more when I'm talking about viruses, but no, they think COVID-19 is a newly formed virus that, although they don't know exactly when it emerged, but it came about recently. It may have been the last couple of years, it may have been the last 20 years, but in the time frame of human medicine, it's been a recent virus. So maybe 20 years ago, or let's say 40 years ago, that virus did not exist on the planet. And I will talk more about that later in the term. Okay?
okay.
They believe it was introduced into humans very recently in 2019, and that they believe it spread from an animal to a human. Now, exactly what happened, we may not ever know, but they have sequenced the virus, and it is similar to 3 other viruses, and they think that this virus recombined 3 times, meaning it was one virus recombined with another virus, and then that virus recombined with a 3rd virus to make COVID-19. And like I said, I'll talk about that when I'm talking about viruses. All right, let's talk a little bit more about emerging infectious diseases. You do need to know the definition of an emerging infectious disease. It's covered in Chapter 1 and a little bit of Chapter 14. Emerging infectious diseases are new or changing diseases, or diseases that are increasing in incidence in the recent past, or are showing a potential to increase in the near future. An example of an emerging infectious disease that they've been talking about for quite a while, and they were afraid it was going to come about, was the bird flu. The bird flu spreads very easily among birds, and then can spread from birds to people, but it doesn't spread much, if at all, from people to people. And they were worried that the virus would change, and then the bird flu would become an emerging infectious disease. If that were to happen, it would be a disaster, because the bird flu is highly lethal. I don't know the lethality numbers, but it's way over 1%. Something like 5 to 10% of people die from the bird flu. Fortunately, that has never happened. The bird flu does not spread among humans, and it has not changed to do so. So hopefully it'll never become an emerging infectious disease. But they're talking about it because it might have the potential to increase in the near future. An emerging infectious disease can be caused by any category of microbe. It can be caused by viruses, bacteria, fungi, protozoa, elements, meaning worms, and by prions. There are various factors that can contribute to the emergence of an EID. These factors include, but are not limited to, microbial adaptation or mutation. For example, if the bird flu adapts to living in the human population and spreading in the human population, it would be an emerging infectious disease. The virus would probably have to have several mutations to do that. At the moment, the virus only spreads, at least easily, among birds. And fortunately, human flu tends to be airborne, meaning we breathe it in. The bird flu spreads mostly among birds by following the fecal to oral route. Any questions about that? And so it would have to change and probably mutate to spread among humans. Global human travel can spread the emergence of an emerging infectious disease. In fact, COVID-19 spread worldwide because of human travel. If there hadn't been any travel between China and the rest of the world, COVID-19 probably would have never come out of China. And it is known now that the reason why it came out of China was that so many cases of COVID-19, the patients are asymptomatic. They have no signs or symptom of disease. And so it was brought into this country, even though we had checks. Like they were checking people coming into the country with a thermometer, and if they were sick, they were put in quarantine. That wasn't very effective because COVID-19 does not always give a fever, even if you have signs and symptoms of COVID-19. And then among young people, I think I've stated this before, about 50% of young people will get infected with COVID-19 and they will have no signs or symptoms of disease. So they won't know they have COVID-19, and yet they can spread COVID-19 to other people. I'm sort of on this COVID-19 spiel here, I apologize. Humans moving into previously uninhabited locations can spread an emerging infectious disease. There was a disease in the forests of, what's that island called after Africa? I'm drawing a blank here. The large island off the east coast of Africa is...
Madagascar?
Yeah, Madagascar, thank you. People started moving into the forests of Madagascar and they picked up a disease, which then spread among people in Madagascar. Fortunately, that disease didn't go further. It stayed localized to Madagascar, and then the government was able to control it. And then global climate change can spread the emerging infectious disease. For example, because of global warming, mosquitoes are spreading further and further north, And because of that, there's real concern that malaria will come back to the United States. If you don't know, malaria was on the East Coast and the southern United States, meaning Texas, Alabama, Mississippi. It went up the Mississippi River pretty far. I don't remember how far. I've got a map of that, but I'm not going to show it. Anyways, the point is, with global climate change, diseases can spread. And malaria is drifting further and further north in Mexico. We got rid of malaria in the United States largely by draining the swamps, reducing the mosquitoes, and then reducing the number of malaria patients. And then with the development of DDT and anti-malaria drugs, we've kept malaria out of this country. It did come back in Louisiana in the 1960s or the 70s. And what they did was they insecticide the entire city. I think it would have had in Louisiana, what's that big city called? Whatever that big city is in Louisiana.
Baton Rouge.
Baton Rouge, thank you. I think they insecticide the whole city and they killed the mosquitoes and then stopped the spread of malaria. All right, are there any questions about emerging infectious diseases? There are a few more slides here, but I'm not going to cover them. With emerging infectious diseases, I mainly want you to know the definition of an emerging infectious disease. Okay? Any questions about that? If there's no questions, I've gotten to the point where I wanted to today, so I think we're going to stop a little early. And I like stopping early anyways because I'm not sure how the Panapto will handle if it's still working on this lesson, and then I want to start the lab. So that's why I give it at least 20 minutes between lessons. All right, any questions? If not, I'll see you at 2:30 for the lab.